This is Boston University's Neurophotonics podcast. My name is Sung An, a third-year PhD candidate in Biomedical Engineering, and I am the producer, editor, and a co-host of this episode of the podcast. This episode, we talk about our first-year experience at Boston University, specifically as a Biomedical Engineering PhD student. We talk about everything from the classes to rotating through different labs to the qualification exam, and qualitatively describe the experience for the graduate program for neuroscience as told to us from friends in the program. That said, thank you for tuning in, and please enjoy the episode.
Hello, everyone. Welcome back to another episode of the Neurophotonics podcast. My name is Dora, and I'm here today with
Sung. I am usually the editor and the man behind the camera, but for various reasons, I am on camera today.
Yeah, well, welcome, Sung. I hope we heard your voice last episode. You were the one who was editing it.
Yes.
We had a lot of fun with this outline in this season of just us chatting about various things. Last episode we covered how we ended up at BU, how we were what type of research we were doing beforehand, and then today we wanted to cover a little bit the initial first year of our PhD, how everything is set up, the classes we were taking, how the rotation system works in our department, and then at the end we will talk a little bit about the qualifying exams because those are a big pain point for all of the incoming PhD students.
Yeah, it's definitely a big kind of landmark moment during our PhD careers. At the very least, it's one of the biggest moments in the first year. So we'll talk a lot more about that later on.
Yeah, but I guess before we dive into the today's topic, we were talking before recording a little bit about us preparing for SFN. Sung, you were telling us about how you're making your poster. I'm also trying to put something together. We got into discussion about font sizes in the end, but it just this is just a nice opening segue into how us as PhD students have just so many different jobs within our one job of basically going from an assistant with scheduling stuff, a technician when we're gathering data, analyst, and then at the end we might even become some designers when we're making our posters.
Or podcasters, who knows,
honestly,
right?
at this point.
SFN, if you don't know what that is Society for Neuroscience. That is the biggest society that deals with neuroscience. So the conference is a bit of a big deal. Especially if you are studying or researching within the field of neuroscience. So, like you said, there's just so many things about being a PhD student, being a PhD candidate that goes into your everyday life. It just all kinds of bleeds together. Not only are you handling experiments, but you're also handling data analysis and interpretation of the results. And more importantly, for the conference, you want to present the results in a clear and nice manner,
Yeah, it is just so interesting when we are spending the majority of our days dealing with very detailed stuff. You have to make sure that everything is set up correctly for your recordings. You have to make sure all of your steps in your data analysis are correct. But at the end of the day, the data that you're presenting, you're talking about very high level stuff. So you have to kind of balance going from this micro scale to the macro scale in that sense. And it's just a very nice, exercise for our brains, which, proves that it's never boring during the PhD. Yeah,
it certainly is. It very much is a creative process, in my opinion. You have all of this work that you are doing to get to a point where you can explain things simply. And I find that to be one of the most challenging things in PhD, is that you have to know the ins and outs of every nuanced detail of the experiment, of the way you're doing the analysis. You got to make sure you understand every line of code that you're putting in. And then at the end of the day, you're spending bulk of the time not really talking about that. You're talking about the nice plots, that hopefully nice plots that you come up with.
Yeah, and they're proving whichever hypothesis you came up with. But yeah, I guess let's take a step back and go back to before us doing research here at BU, coming in as a first year. So in the biomedical engineering department, which all three of us are part of, when you come in your first year, depending on whether you're a post-bachelor or post-master student, you have various course requirements. So for example, for me, I was a post-master student, so I only had to take courses in my first year. There are a few courses that basically everybody from your cohort takes, which include
605?
605, which is some type of synthetic biology, and then 606, which was physiology.
Quantitative
physiology.
Yeah.
So, and also the math requirements.
Yes.
Which are definitely fast-paced, to put it so.
Yeah, it seemed like, I don't know if it was the same for your cohort, but in my cohort, most of my cohort basically took the same class for the math requirement, which was, I think, a linear algebra course.
Yeah, and the stats course, that's kind of a 2-in-1 within one semester. Yeah, it was a really nice, I think, bonding experience for our cohort because we were all doing the homeworks together. We were trying to problem solve together, and that's how you basically, got to know the people you were working with, but also got adjusted to this new system that you got put in.
Yeah, and I think that's like a really nice kind of segue into the rest of the PhD career. It's a nice taste of what's to come in that you're really working with people who have the same objectives as you, especially in a class like that. Your objective as a student is not only to learn the topics at hand, but also to complete the assignments, which does feel like a bit of a taste of like what being a PhD student is like and what doing research is like, because you're not only trying to get at the question that you're curious about, but you're also doing the assignment, making papers and creating posters and stuff like that. So I was unfortunately not in the same math class that I was that most of my cohort was in, but I think the core requirement courses do kind of give a nice introduction into BME in general, like the whole wide scope of what BME is.
Yeah.
And Sung, why weren't you in the core requirement math class? Did you already have that background? Is that why you chose another course? Would you want to expand on that?
Yeah, I was a mathematics major back in undergrad, and that meant that most of the math requirements I already took, but I was a theoretical mathematician before coming into BME, which meant that the classes that I was more interested in was more application based, which was why I took a mathematics for engineers or something. I can't remember the exact course title, but that was the class I took.
And do you think that was a useful class into segueing into BME research?
To be honest, it is so far from what I'm doing currently that I'm not entirely sure that that was the course that really helped me along. But there was another course taught by Professor Mark Kramer that looked at neuroscience and modeling in neuroscience, and that was really helpful.
Yeah, it is. I'm also thinking back to the synthetic biology course. I don't know what it was called exactly, the 605 course. And, I mean, from my background, I had more of an EC computer science background. I've never been in a wet lab prior to coming to BU, or not a synthetic biology wet lab. So for me to go in there and pipette stuff and imagine that there is, something slicing the DNA that we're gonna multiply there, it was just basically science fiction. However, I'm still very appreciative of that base knowledge that I gained of, okay, like this is the whole breadth of the field, that there's just so many things that you can work on. And then there's, of course, people who join synthetic biology labs who are still working on very specific topics within that field. And even though I'm now doing computational neuroscience, knowing that background does really help me when I'm reading papers to put everything into context and to, just contextualize it in a broader sense.
Right. Just like a little bit of a tangent on that. When you're doing research, you don't want to just be reading papers from just your own very specific niche topic of interest, right? You want to be reading papers that are all over the place so you can grab ideas from different fields. And having that kind of background knowledge really does help when you're trying to get more different ideas and a variety of ideas to synthesize them into something that you can create, right? And it also helps because after spending a year with your cohort in the rest of your PhD career, you're probably going to be in your labs and doing your specific thing. But as somebody who's looking at neuroscience, it does help to have that common knowledge of, oh, we took that class together and we have that same shared knowledge from that class. And that just kind of allows you to talk with people in your cohort about their research, even if they're not doing the same thing as you.
Yeah, and you just know where to find things.
Yeah, absolutely. Great points. I wanted to highlight the point that Sung just said. More than anything, if you're calling yourself biomedical engineer or like neuroscientist and something, people immediately expect you to have some sort of domain knowledge. So even if you're doing computational neuroscience or, I don't know, more mechanical engineering, mechanobiology, and whatever that you're doing, having the breadth of knowledge in biomedical engineering core principles like DNA, PCR, and having some wet lab as to talk with researchers outside your research areas, but within the biomedical engineering domain is useful. And to chat with your cohort is always fun about that. And thanks for talking about your mathematical background. Another example of how interdisciplinary NPC can get.
Yeah.
Well, so on top of taking classes, people usually have to balance rotations as well.
Right.
And in the biomedical engineering department, there are a few subsections, I would say. So there is the synthetic biology little group. There's the neuroscience based sub cohort to call it like that. There's professors who are focusing on optics. So usually students are encouraged to rotate with whichever lab they want. There is absolutely no restrictions. However, what tends to be the pattern is that students come in with a specific area they would like to focus on. So for example, for me, I knew I wanted to get into neuroscience. So my first rotation was more an optics side of it. So I was actually rotating with Professor Biggio, who we interviewed recently, and I was looking at all of those myelinated sheets, and I was learning how to use a microscope for sliced tissue. And then my second rotation was more experimental neuroscience in that sense that I was working with mice. I was doing imaging, and then I transitioned into the computational side of things. So that was, for example, my path. But
Yeah, usually people have an inkling of what they want to be studying for the rest of their careers. And to be completely transparent, they do talk about qualifying exams when you are entering the program kind of off the bat. And the qualifying exams have 6 different subjects to them. Was it 5 or 6?
I can name 4 right now.
Yeah. Some of them are the ones that have already been mentioned, like optics and neuroscience.
Computational something, but biomechanics is one of the things that we forgot to mention.
Tissue engineering as well, I think, might be one of them. So there's roughly 5, maybe 6.
I think those were all the ones. And if we're forgetting, we'll put it in the description.
Yeah.
Yeah, and then brings back to the point. Even if you're doing computational quals, let's say, and if there is a computational RNA technology, or let's say the Google's DeepMind AlphaFold, you need to have the domain knowledge to even comprehend that data, right? You can't go into the computational quals and not know what RNA is or DNA is, because they'll ask the question. You have to know the domain. So that's why definitely, even if it's not in your domain, definitely helpful to take all those core courses.
Yeah, we'll talk more about this later, obviously. But there are significant overlaps between the different topics, and having not just one area of expertise really helps you along with understanding the papers, right? So when choosing rotations, at least for me, I was also keeping in mind, okay, what are the topics that I want to be choosing for my qualifying exam, and which rotation labs are going to be helpful in gaining kind of hands-on knowledge to understand the papers.
That's interesting. I was never thinking about my quals so far in advance. Maybe because I already knew what I didn't want to do, so that left me with just the two topics that I wanted to get into. So it was never a big questionable thing of, okay, what do I want to do?
Yeah.
But yeah, I mean, I remember people fighting over who's going to rotate with whom, and just scheduling those rotations, figuring out who has space in the lab. So I guess that's, I guess, an admin thing that is worth mentioning, that sometimes it's not you who is an unfit person for the lab. I mean, please do not doubt your skills. Sometimes it's just that certain labs are not taking on students. So what's good about the department here is that professors who cannot take on students into their labs are not going to mostly allow you even to do a rotation with them. So that way you are not wasting your own time, and you will be able to explore other avenues you might be interested in.
Yeah, to add on that, but at the same time, even if the professors are not taking students, and if you have already found the research home, to gain more knowledge and expertise, you can always choose to rotate with professors. And in those type of cases, professors are open to train you for a rotation project, even knowing that they're not going to join the lab. So that's also an option.
Yeah, and that's always nice to establish collaborators in that sense, because professors are sometimes looking into avenues of collaborating with other professors, and then you might be that middleman, middlewoman, that link between the two research labs, because you have the interest in the two expertises these labs are offering. So it's always nice to just like expand, get out of your comfort zone, and explore.
Yeah, it really is. And kind of touching back to something that you said before, is that you don't want to be wasting time with these rotations. And yes, the rotations do take time. In your first year, you're not just working on assignments, you're also working on rotation projects in the labs. And it's not going to be as simple as, here, I'm going to throw you a bunch of code and data and you get to analyze it or whatever. And for some, even that might be like a lot to take in, right? But yeah, it takes time. It takes energy and effort in order to learn things at the rotations.
Yeah, I think it depends very much on the lab and on the project you have been assigned to, and also how interested you are in the lab to begin with. Because sometimes you realize 2 weeks in that you're not interested in something, you can, then focus on your coursework maybe a little bit more, and then start your next rotation a little bit early. What I wanted to say is that professors are also very understanding with us as students taking classes, just because classes are something that you need to get out of the way basically immediately. So there's never a strong emphasis on the rotation project. It's not about the science of the rotation project. It's more about you getting to know the lab, the techniques that they're using, and just the general lab environment, and see if that would be something that you would enjoy doing.
Yeah, that is an important point, because your rotation doesn't necessarily depend on the success of your rotation project. It's more about, do you fit in well with the lab culture? Do you fit in well with the way that the lab is being run? Of course, assuming that rotations are a good representation of what the lab is running like. If that suits you well, or if that doesn't suit you well, that would be a major factor in you trying to figure out and navigate how to find your research home.
Yeah, I guess we can touch a little bit more on the second part of the first year of, okay, the first semester is usually overwhelming with with figuring out classes and starting the rotations. But I feel like starting the spring semester, you already get into a rhythm. You know how the classes work. You know how to be successful at them. You're slowly wrapping up, finding a research home, and then you have the next step, which is the qualifying exams. So do you maybe remember, or can you tell us a little bit more about how your cohort approached the qualifying exams? When did you guys start preparing for it, or how did the process look in general?
Okay, let's talk about that beast. So to be honest, it becomes kind of a blur after the fact. So I can't give you exact timelines. I can't tell you like, oh, on like April 28th, we got our papers and we're gonna start looking at them. And then people don't look at them until like 2 weeks before the actual exam. But that is actually not the approach we took, but I'm just taking that as for example. I think for us, what was interesting was we had a bunch of people that were interested in the neuroscience topic and the computational topics. But then we had like 5 people interested in optics, and I was interested in optics, Neurophotonics Center. Because of that, there were the 5 people that were interested in optics, and 4 out of the 5 people were interested in neuroscience as well. So we had 4 people that would meet regularly earlier to discuss the neuroscience papers, and then the 5th one would come along later, and then we talk about the optics papers. So each day was like 5-6 hours of just looking over the papers together, making sure we're understanding the topics and trying to figure out, are we really understanding it, or is this feeling like we're just understanding it because we've been talking about it for the past 2 weeks?
Yeah, fair enough. So just to put a pin in there, you mentioned papers. So to give a little bit more information, every topic that you choose, so whether if it's neuroscience, whether if it's optics, every student chooses 2 topics. So for the 2 of us, it was, or in Sudan as well, it was optics and neuroscience. And then certain professors, I think they're rotating every year, select 3 papers in total for each topic that you read about and that you present later to your committee. And then for example, for our neuroscience papers, we had a very nice variety of going from a neuroscience topic that was very systems neuroscience focused on silencing certain cells, like looking at circuits. We had a paper that was basically synth bio with like all of the cell imaging techniques. I don't even know. Like I remember that there was some type of clustering over there. And then the 3rd paper was machine learning, fMRI, analysis of data. So it was a really nice variety, and it's still not just the papers. You actually have to understand the background of like what context did these papers originate from, like why were they written.
Yeah. Just as a disclaimer, we are all, again, BME students, so we'll be talking more about the BME quals. But I believe the GPN, which is the graduate program for neuroscience, I believe they do quals slightly differently. We can touch a little bit on that qualitatively, but for now, yes, we do get 3 papers. And us as well, we got a really nice range for the neuroscience papers. I think one of them was about plasticity, one of them was about neurostimulation. And I think what really helps is that because there's so many different topics available in neuroscience, and yeah, because it's from such a wide range of different topics, the way the quals are carried out, you are not necessarily asked about all 3 of the papers. Sometimes people only get through 2 or maybe even just 1. And the committee is from anecdotal experiences. Most of the time, it seems like the committee is fairly flexible. If you feel like you're uncomfortable talking about a specific paper for whatever reason, they will allow you to start moving on to the next paper. And Should we talk a little bit about how the committee is chosen as well? Like what that is like, the experience?
So your committee for quals consists of 3 people. One is the chair, and the role of the chair is basically just to observe and see if the 2 other committee members, both of which are in charge of a certain topic. So for us, it was neuroscience or optics. You have a professor who is an expert in the field. They come in and then they question you. And of course, other committee members are chiming in. It's a conversation just because everything is so interdisciplinary and everybody has opinions and it's more of a discussion. And I think it's more about assessing your critical reasoning and your potential to do independent research rather than you remembering every single thing that has been done in set papers.
And that's a very important point, right? It's meant to get you to the boundaries of what you are comfortable with. And that is something that I did really appreciate a lot about the quals, is that it made me confront the extent of my knowledge. And they're very, very good at least in my experience, they're very good at getting you to a point, leading you with follow-up questions to a point where you can not really know for sure because that's not within your expertise yet, but you can reasonably make critical reasoning assumptions to get to a point where, hey, I even learned some stuff while taking the quals. You know?
For sure. You remember that.
Yeah, exactly. And yeah, at the moment, it's a lot of pressure because 3 people are there and they're staring at you. And at least it feels like they're assessing how intelligent you are. But that's not really what they are looking for.
No, they're just looking if you can work well under pressure. They're just basically condensing the PhD experience into this 1-hour session of presenting research, defending that research, or taking it apart. For example, for one of the papers that I was being questioned on, it was not the best paper. It definitely had some loopholes in it, and because my research project was supposed to be very similar to that topic, I actually went and tore apart that paper, and we had a really nice discussion on why I thought that the paper was not designed well, why I thought that it could have been done better in certain aspects, and then we went back to the basics. So that was like the fMRI machine learning paper. And we went back into some machine learning algorithms and why I thought that certain other algorithms should have been used or differently designed cohorts for the data, et cetera, et cetera. So it was a very very open discussion. And for me, it was a very nice experience, honestly.
Yeah, it is not to take anything away from the experience, but yes, it was super stressful, but
for sure.
In hindsight, I really appreciated and I really enjoyed that time of not only the qual itself, but the moments leading up to the quals, becoming a close-knit community with the people who will eventually be doing the same things as you are, because that's just how the quals are designed. You're looking at the topics that you will be looking at for the rest of your career.
Yeah, I just didn't appreciate I was in the last group on the last day. So basically quals start on a Monday and I was somewhere Wednesday afternoon. So starting Monday, I was not coming into the office because there were already people who have gotten through with it and were, just very happy and relaxed. And I was definitely not relaxed at that point. So that is definitely something to keep in mind. Yeah. But, it passed. It was great. And also one other thing, in case something happens during quals, the department does allow you to retake the quals. So it's definitely not set up for just to see you fail. It's not set up in a malicious way. So you definitely do get a second chance in case you were anxious, in case something happened and you couldn't prepare and present in the way you wanted to.
Yeah, they want you to succeed. And it might not feel like it, but they do definitely want you to succeed. And I think that's something that I really did feel while taking the quals as well. The questions that were being asked were getting to the point where it seemed like they knew that I didn't know about something. So they wanted me to get there on my own by asking those questions. And that was really cool. One thing that we probably should talk about also is that, yes, the quals are separated out into 3 different days. And obviously, these professors are coming in with biases and like a certain level of comfort in their expertise as well. So what they also ask you to do at the very beginning is to first, talk about the rotations that you've been through and the classes you've taken. Maybe I think that might have been also it, right? Your background, like what your undergrad experience was like, and then the chair is also there to make sure that between all these different committees, the experience is as uniform as possible.
Yeah, I think that's all in terms of quals that I remember and that is worth mentioning at this point. One last thing that is not quals directly like quals related is that we took a cohort post-quals trip,
Yes, yeah.
which I think we mentioned last time. Where actually Sudan and I became friends and we started talking. So that was also a really nice way of just celebrating the end of the first year of the PhD and preparing for our first summer of research.
Yeah, that was a blast. We went to a place in Maine. It was so outside of the city and it was just like in the middle of nowhere. And that was honestly one of the most fun I've had in that first year of getting to meet people, because you just kind of get to know people outside of research, outside of this spatial environment we call Boston University. You're taken to a different location, and in that different context, you get to meet your cohort and share in the experience of having completed this kind of giant beast, really, that looms over us for the entire first year.
Right.
And
Yeah.
we come out of that experience having bonded a lot more.
Yeah.
Yeah.
Yeah, for sure. For us, it was also a really nice trip. We went to Rhode Island. Definitely nobody was anybody swimming at that point? It was June, but I think it was
Rocky definitely was swimming.
Shout out, Rocky.
Should we talk very briefly about the GPN process, or what we know of the GPN process, and then wrap it up?
Sure. I mean, I can attest anecdotally to the bioinformatics department, because that's what I have a friend there, and they do have a written and oral examination. It's not based on papers, but it is based on topics. So I would say that it's somewhat similar. It is dragged out a little bit because I think they take their written one in the fall of their second year, and then the oral qualifying exam somewhere in their third year. So it is a little bit of a longer process in that sense. And then what I also know is that some other departments call their prospectuses qualifying exams, which I think we will touch on in our next non-interview episode, where you actually put together a committee who will be the committee when you are defending your dissertation, and that's your quals are basically your first time presenting what you plan to do for your dissertation. So that is sometimes called quals depending on the department, and then in the BME department that's called the prospectus.
Yeah, it does seem like, from talking with different departments and people that went to different grad schools, that we are fairly, maybe not unique, but different from the norm of taking the quals at the end of the second year. Yeah, at the end of the second year is when most people seem to be taking their quals, which makes it interesting that we become PhD candidates after first year.
Right, because
away.
that is the big distinction, that you're a PhD student until you have taken your qualifying exam, and then you become a PhD candidate.
Yes, yeah. So I do think that is interesting, but I do also really appreciate it, because not only do we get this 1 year of rotating through different labs, we don't come in knowing which lab we're going to be participating in for the rest of our career right away. we come in and we get to experience these different labs, different lab cultures, what big labs are like, what small labs are like, what starting labs are like, and what established labs are like. We also get this added benefit of having this qualifying exam, seeing where the extent of our knowledge lies, and then realizing how best to supplement that and what courses we could take in the second year if we want to take courses in the second year to supplement that knowledge and then become a better researcher, a better student. So I think that's a really nice way that at least BME has set it up for.
Yeah, it has been really enjoyable. And then one other thing we could talk about in one of the next episodes is teaching as well.
Oh,
because
yes.
that's something we have completely forgotten about for some reason when we were making our notes.
Yeah, second
but
and
that
third
is,
year, yeah.
Yeah, that is definitely an experience in and of itself. And also is just building a skill set that can be utilized later on.
So shall we wrap it up? I mean, this episode, we don't know if I'm the editor here. I am also preparing for SFN. I don't know if I'll be able to finish this episode and have it uploaded before SFN. But in the case that it does, if people want to come say hi to us at SFN, I believe all three of us are going.
Yeah.
So that's going to be fun. If this comes out after SFN, hopefully we met you at SFN. I don't know. Yeah.
Yeah, and then we will have probably an interviewee next time.
Yeah.
And so that will be, again, a nice segue to go back into the more science-heavy episodes. So yeah, I think it will be fun, and we have a really nice plan for this year. I'm really excited.
Yeah, this season's going to be great. We'll still keep the interview pretty easy breezy, right?
Yes, definitely.
Yeah.
Okay, well, thank you so much for joining us for this episode of the Neurophotonics podcast, and we hope to see you next time.
Yeah. Bye-bye.
Bye.
Thank you for listening. If you like this episode and want more like it, please take a moment to share it with anyone you think would enjoy it too. If you are listening on Spotify, please check out our YouTube version of the episode for some bloopers and outtakes. If you are watching on YouTube, these will follow shortly after the following message. This episode was made possible by the support of Boston University's Neurophotonics Center and listeners like you. Thank you. I mean, the fact that billboards are still a thing baffles me. I think.
And on top of that, one of the advertisements says "Drive safe."
Oh yeah. Or
Wait, my favorite one is I got hurt in. But by the way, no free advertisements. No free advertisements for them.
one of my favorites is like, did you get into an accident? Yeah, I was reading the billboard.
You're at like 123B,
Yeah.
and you know exactly
Yeah.
which exit you have to take.
You didn't have to do that.
Yeah, those are my grievances with the American traffic system. Anyway, hello.
The qualling? Does anybody say qualling?
I don't
feel
think
like
so.
that's a natural way to call it, though. Okay, what should we talk about next?
Usually people have. I don't know what I wanted to say.
How's the bottom of my shoe looking?
Ooh, that's the cleanest.
How are my slides looking?
Your slides? Oh, are those called slides?
Well, Birkenstocks. How are my Birks looking?
I don't know, Dora. When you said slides, I'm such a nerd. I thought you were talking about like Hopwin slides.
That's what I thought too.
I cannot make my title 115 PT. Like what? Like what do you mean?
I just slide it around until it looks nice.
Okay, fair enough.
I don't know who that is.
You gotta come to more socials.
And today, my name is. Oh, what? Yeah.
Today your name is.
Today. Okay, let's restart.
Let's restart.
Sorry.
Sit back and kind of look at all these pretty pictures that come in.
Oh my God, yes, I still have it as my LinkedIn cover photo.
Oh yeah. Want to see more of those pictures? You can watch that episode.
We'll put the link in the description.
Okay,
Oops.
are we starting the recording today?
Did we start the recording? I mean, you should have told us that you were starting recording. Just saying.
Just saying.
I mean,
Wow.
that's not very ethical.
Such a
Maybe you'll be the cameraman from now on if you do this well. We'll see what big labs are like, what small labs are like, what starting labs are like, and what established labs are like. It's like a Dr. Seuss thing. Do the
Just say
thing, say bye. Just come.
Sudan, say bye.
Bye.